So now we're ready to make the gel, and there are lab procedures in your manual as well as instructions online at the Rutgers site. So you're going to get the agarose jar from the cabinet, and we're going to label it. Make sure you label it with not only 1% gel but your initials, so that I know who made the agarose. And you're going to label this jar because you're not going to use the entire 100 milliliters of agarose. So you're going to make a 1 gram. You're going to take 1 gram of agarose measured on a balance, and you're going to add 50 milliliters of buffer, not water. Make sure it's TAE buffer and that we've made it in the lab. If you don't find any TAE buffer, we have concentrated 50x TAE that you can dilute, but there are instructions to do that as well. So make sure you talk to me or one of the other students. You're going to add that to your agarose, and you're going to swirl gently, and don't let the agarose get up into the neck of the jar. You want it to stay at the bottom. You're going to microwave it in 30-second increments until it melts. And again, I mentioned this in lab. You want to make sure that it melts clear, and you need to have your hot hands from the cabinet ready, because this bottle is going to get very hot, and the agarose can actually start to bubble and melt over and out of the jar if you're not careful. So 30-second increments, just to make sure you don't boil it over. If you do boil it over, just make sure there's paper towel in the microwave before you start doing this, so that if it does boil over, you can just throw out the paper towel and not leave a mess. Once it's clear and you have no little specks in the agarose, it's completely clear, you're going to add the other 50 milliliters of buffer to the melted agarose and swirl to mix. This will cool it down enough so that you can pour it. You're going to give it to me so I can add ethidium bromide, and then you're going to tell me which gel box you're going to pour the sample in, and you're going to do this and make sure that the casting tray, if you're using the gel boxes that have separate casting trays, you're going to make sure the casting trays are taped and that you have a comb in there that with the right number of wells. You're also going to put a piece of tape in front of that gel with your name on it, so that when it's done, you're going to pop it into a gel box with buffer, and once it's solidified and it's in buffer, you can leave it until the next time you're going to use it. Generally, it takes about 15 minutes to solidify, but it depends on how hot the agarose is. Once you're ready to load your samples, you're going to load 2.5 microliters of the gel loading dye into the PCR tube. And the loading dye doesn't allow us to see the DNA. It allows us to see the sample and to be able to tell where we're loading the sample. So again, the only thing that's going to help us visualize the DNA on it in the gel is the ethidium bromide that's going to fluoresce when you shine it with UV light. In the loading dye, you have these 3 components, and again, it's to help us visualize the sample and be able to see where we're loading it. And these 2 dyes are both negatively charged and both run towards the positive end of the box. The bromophenol blue is smaller, and so it'll run faster. The glycerol in this loading dye makes the sample sink, so we're able to load our sample and it sinks into the wells. So there is a video that should be linked to your site, to your classroom, and that video shows you different types of techniques and errors that may occur when loading samples in gels. So it's a good idea to watch it to see what could happen next time when you load a sample in a gel. So how does the agarose gel work? The agarose itself is a sugar that forms pores and creates these little tiny particles that create channels in the agarose. When you load the sample, you're going to pull the negatively charged DNA particles through the agarose, and the larger they are, the harder it is to travel through. So the tiny pieces, the small pieces, run fast, and they will run at a different rate. So that allows us to separate the pieces of DNA based on size and because they're charged. Again, when you do separate it, we're actually going to run a ladder or a marker in your gel to be able to see how large your PCR sample is. And again, we're going to have a PCR sample, our actual insert, the fragment that's here is different than the gene insert. The gene insert is only the area of the gene that we're looking for. The fragment is going to have about 200 base pairs extra that come from the original vector. Okay, so let's take a look at the mocked up versions of an agarose gel, and we were working on this in the lab. Basically, you're going to put your name and the samples and the numbers, and you're going to load a marker into your well, and then you're going to load your samples. By loading a marker later, we're able to see the size of the sample. And again, we have a cutoff, a minimum cutoff for our gels, and that if they're smaller than that, we typically don't want to proceed. But for our purposes, we haven't had that problem, so we usually run through the labs anyway. Okay, so a couple examples here. Are the markers visible? Did the gel run okay? Does it look at it like it's as expected? Basically, in this case, it looks pretty messy here. And what could have done this? Basically, if you see these streak patterns where there's white in between your sample, what could have happened is that you didn't melt the agarose completely and there were tiny particles of agarose in the gel. So when the sample runs through, you hit these pieces that the DNA didn't flow through, so it appears lighter. So again, this happens if you have particles or little pieces of agarose that are not completely melted. But in this case, I can tell the size of the fragments because my ladder is visible and I can see the segments of the ladder. So you could still use this data, it's just not very pretty. Okay, did the PCR work? Are the bands, are there bands? Yeah, if you see the markers there and there are bands, yes, the PCR worked. Okay, and if you take a look at lane AV3 right here, you see that there's more than one band. In PCR, you're amplifying a fragment, a section of your plasmid, and there should only be one section. There should not be more than one. If it's more than one band, that means that you amplified two different plasmids. So there could be a contamination in your gel. Or could it be possible that the person next to you did their sample kind of leak into your well? Okay, it's possible. That's why we continue and we analyze. We continue to do the processes in the different labs to analyze that sample. So what is the size of the insert? Again, I told you there's an extra 200, right? So if we look at this one, we have we have an insert here, but it's at 200. That means that maybe we didn't have an insert at all. No, we didn't, because you're only getting that section here and here. There's no in between, which means that it just amplified that little section of 200 bases. So if you get a fragment of 200, we subtract 200. So that means that there's 0 base pairs. Okay, and so let's take a look at the size of the JM1 insert. Here you have an insert or a fragment that's about 650, maybe a little less. And again, we subtract it, we get about 400. So here's your clicker question. What is the size of JM2.10 insert? What is the size of this insert? And last, I would like you to print out, go to the original PowerPoint and print out this page and answer these questions and hand them in during class next week and during our first meeting on Wednesday. If you have any questions, please email me and we can discuss this in class. Have a nice day. Oh, by the way, before I forget, there are these 2 links that will be found on the classroom and in EDU 2.0. Some of them are just funny videos that different companies have put out about PCR. But this one, the CSI, is interesting because they basically, I want you to watch the video and the video clip and just see how quickly can they do PCR. And I want you to think about how long it took you to do PCR and get your samples back. So have a nice day, everyone. If you have any questions, please email.